Hello, I'm Gavi.
I'm Gauri.
And I'm Ashley, and today we're going to give you our perspective on the power of antibiotic resistance.
Now, bacteria are all around us. Bacteria can be both good or bad. Digestive bacteria are good, pathogenic bacteria, for example, are bad. Now, antibiotic resistance is becoming a major threat. For example, as shown by this graphic, MRSA is antibiotic resistant and it continues evolving and becoming more resistant.
So we see antibiotics as this major force against bacteria. But what do antibiotics do? How do they work? So there are two different types. There are beta-lactams, which mostly affect gram-positive bacteria because they fight an enzyme called transpeptidase, which helps form the peptidoglycan in their cell wall. Examples of beta-lactams are penicillin and ampicillin. So as you can see here, they tear down the cell wall, the peptidoglycan. Aminoglycosides, on the other hand, they affect protein synthesis. They're mostly effective against gram-negative bacteria. So as you can see here, we have our typical bacteria, and they work by going against the ribosome. So, wow, this video is taking a long time. So as you can see here, protein synthesis in the ribosome, so translation of the mRNA sequence. So an amino acid is forming, and as it's going further and further, an antibiotic like streptomycin, which inhibits translation, will come in and stop translation and protein synthesis.
So, how do bacteria grow? Well, there are 4 phases of bacterial growth: the lag phase, the exponential phase, the stationary phase, and the death phase. So, first there's a lag phase where there is a relatively low number of bacteria. Then, once there are enough bacteria, the exponential phase occurs, in which the number of bacteria really start to rise. After this is over, in the stationary phase, the number of bacteria reach a certain threshold and stop increasing in number. And in the death phase, they begin to die off.
Now, mutations are very important. There are 2 models of mutations. There's the induced model for mutations and a spontaneous mutation model. The induced mutation model says that bacteria get mutations in response to environmental pressures, like the introduction of an antibiotic. For the spontaneous model, bacteria get mutations randomly, not necessarily due to environmental pressures. Therefore, the bacteria line accumulates a lot of mutations across a whole span of generations in the spontaneous model, while in the induced model, the mutations only arise in the one affected generation. Therefore, the spontaneous model is the correct one, according to Luria and Delbruck. So therefore, mutations occur spontaneously.
So, why do we use Serratia marcescens in our lab? Well, first of all, it's usually not pathogenic, so it's not likely to harm you. Also, it is easy to grow, and some bacteria have a really bright red pigmentations, which makes them easy to see and count and analyze.
So now let's pull everything together. What's the role with bacteria and antibiotics, and how do populations of bacteria become resistant? So the short answer is selection. So spontaneous mutations give bacteria an edge. So if one bacteria has antibiotic resistance, it's going to propagate and grow because it can fight the antibiotic. As more populations grow, the antibiotic becomes less and less effective, and that's where we have a problem with mass epidemics of bacteria where we can't necessarily fight. And as you can see here, it's kind of dangerous because they can reproduce very quickly through conjugation and other reproductive methods.
So, what are we doing to stop it? Well, there's a lot of research on this. Some methods to combat resistant bacteria are the development of new antibiotics as well as phage therapy. This is the use of bacteriophages, which are viruses that parasitize bacterium to treat pathogenic bacterial infections. There are also clinical treatments. There are vaccines to prevent infections. There's the awareness, raising public awareness, and there's also the prevention of the unnecessary use of antibiotics.
So thank you. We hope you
Enjoyed our
view
view
on
on the power of resistance.